Unveiling a promising pathway, targeting ABHD18 for treating Barth syndrome. When it comes to battling rare genetic disorders, every breakthrough feels like finding a needle in a haystack. But here's where it gets intriguing. A recent study published in Nature might have just uncovered a game-changing strategy for managing Barth syndrome. This rare X-linked condition, primarily affecting boys, stems from a mutation in the Tafazzin gene, leading to a severe deficiency of cardiolipin, Cl, a crucial phospholipid for mitochondrial health, and an alarming surplus of monolysocardiolipin, MLCl, a detrimental lipid that wreaks havoc on mitochondrial function. Individuals living with Barth syndrome often endure a harrowing journey, marked by muscle weakness, recurrent infections, and cardiomyopathy. Despite heart transplants offering some respite for cardiac issues, the prognosis remains grim, with many children failing to reach adulthood. Yet, amidst the challenges, there's a beacon of hope. An international collaborative effort involving researchers from the Hospital for Sick Children, the University of Toronto, and other esteemed institutions has identified a potential therapeutic target that could revolutionize the treatment landscape for this devastating condition. Enter ABHD18, a previously enigmatic gene whose function was largely unknown until now. According to Sana Massoud, a former PhD candidate under the guidance of Jason Moffat, PhD, a senior scientist and program head for genetics and genome biology at the Hospital for Sick Children. The team's initial screenings for genes interacting with Tafazzin led to an unexpected revelation. We found that ABHD18 encodes a pivotal regulator of Cl metabolism, she shared. This means that ABHD18 plays a critical role in converting Cl into MLCl, a process that exacerbates the condition when uncontrolled. By inhibiting ABHD18, the scientists observed remarkable improvements in mitochondrial health and heart function in preclinical models. It's one of the most striking examples of a disease modifier we've encountered, remarked Vincent Bloman, PhD, co-lead author of the study and senior director of discovery sciences at Scenic Biotech. Using a small molecule inhibitor named ABD646, they were able to significantly lower MLCl levels in various models of Barth syndrome, including zebrafish and patient-derived cells. Jason Moffat, also a professor of molecular genetics at the University of Toronto, sees this discovery as a direct route to alleviating the root cause of Barth syndrome. This breakthrough offers genuine hope to patients and their families, demonstrating how cutting-edge research can pave the way for novel treatments and improved care for those affected by even the rarest of diseases. And this is the part most people miss. While the focus remains on understanding the intricate mechanisms behind ABHD18's regulation of Cl metabolism, the potential implications extend far beyond Barth syndrome. Could targeting ABHD18 open doors to treating other mitochondrial disorders? What are your thoughts on this groundbreaking research? Share your insights in the comments below.